So let me wait a little bit for my slides to appear. Okay. So thank you for coming today. Today we'll be looking at DIY precision medicine with Pandas. So it's DIY not because you should now abandon all of the precision medicine companies. It's more like when you do something and you as a programmer know, you will understand better how the things are working. So that's why it's DIY. So you start up like in the morning, you look at some articles and you see, wow, lifting weights, maybe I should lift weights to be healthy. Or maybe walking, this is the thing that will help me to be fit and healthy. What about doing squats? Maybe this is the goal to be fit. Or avocado, we're opening completely different dimensions of superfood. Or some, I don't know, whatever clickbait about vitamins. And so you think like maybe I should just relax, like I don't know, like should I do anything with myself? Like, so no, you should not. But we can have science to help you with that motivation. So why am I talking about it? I have a background in genetics. I'm an educator, bioinformatician, and data scientist. I'm also a mother of 3 little Zen masters. You will see them tomorrow rumbling around. And that's why my favorite quote is, I like to challenge myself. So are you ready to start with the precision medicine? Okay. So with the traditional medicine, that you all experienced. You come to the doctor make you some prescription, and for some of us it works. As you can see in this case, like the effect is good. For some of us it's like, doesn't do anything, but like doesn't hurt, that's okay. And for some of us it's actually even worse when you took that pill. So what is precision medicine doing? And I repeated this word already like several times, so what is actually precision medicine? So the precision is coming from the data. So you have the same group of patients as in the first example, and you have data from them. So the data could be different data, and by gathering this data, you can divide these patients into multiple groups and have a different therapy based on what group patients are. And by that, you can account to the good effect in each of the patient group. So precision medicine, it's not only about the therapy, because, you know, like, why did I start it? Because all of this, like fitness and so on. But it's also the set of customized recommendation for your lifestyle, for some prevention. Should you go to the doctor every year or every half year or like every 2 years? So this is some of the questions that precision medicine can answer. And so I said that there are different data that precision medicine could use. So what are they? Just few of them. It's activity data. So if you have your Fitbit or whatever you have, this data could be used to make some customized recommendation for you. Also your biochemistry, your vitamins level, your hormones level. Then imaging, looking at your, doing MRI of your brain, for example, looking at how your brain is functioning. And the part that I will be focused on today is the omics part. So, and I will be focusing on genomics. So omics are all of these different fields, like genomics, your DNA, transcriptomics, your RNA, or proteomics, your proteins. So when we look into genomics analysis, and genomic analysis, we need to do that because, remember, we need to actually divide our patients to see are they in a high-risk group or a low-risk group. So what do? First, we select trait. We select what are we going to study in these people. So diabetes, this will be the case of the code that I will be showing today. Or menopause, or any cognitive disorders, any of that trait, or phenotype we call it, you should select it, and you should select specific people for that. So either the people who have it or doesn't have, or have some continuous representation. Then you do some association studies. So there will be some abbreviations here, and I will go through them afterwards. So you do some association, and then you're creating some risk score model. And this seems to be like really fancy, so like all of this genomic stuff. But if you look into that, and I will be actually going into the details, these are like a bunch of different regression models, which are scaled when you use a lot of samples. So we start with the first one, and it's you need to select and create cohorts. So a lot of different words here. So we'll start with a cohort. So cohort is some group that you are selecting based on the data that you have. Usually you would use biobank data. So I'm working in the company called DNA Nexus, and there we are providing data of UK Biobank, and it's a longitudinal study in UK where they have 500,000 people that they are looking into. So we have 500,000 their DNA that we can study. However, the data are not open to everyone, so today I will be showing it in the different data. So you have this data, now you need to define what you are searching for, what phenotype, what trait, and it could be either continuous, for example, blood pressure, or it could be binary, and so blood pressure is continuous, having a high blood pressure is a binary. And based on that, you are creating cohorts. For example, if you have a binary phenotype, you are creating cases and controls. And you would like it to be as balanced as it could, which is not so often, so you need to account for that. Then you are making some data cleaning of your samples. So usually we do not use any relatives, because then the association is, so the frequency then is different, because you got your DNA from your parents, so it's not like just independent occurrence. So that's why we do not want to have relatives in these studies. You also want to have people with the same ethnic backgrounds, again, because these jeopardize your frequency of the variants that you are looking into. And you don't want to have some major chromosomal change, which actually might be a shock for you, but a lot of people have some chromosomal change and they do not know about it, but this could, again, mess with the data. So we want to cover that, and then we want to use this data. So I talk about the biobanks, and there are actually a lot of different initiatives for biobanks. So you can see that these are all of the different biobanks that are available. So I want to point the biobank that I'm working with as a community engagement scientist. It's UK Biobank. It's one of the largest longitudinal biobank. But today, the code that you will be looking into in the second part of this talk will be focused on 1,000 genome project, which actually is not 1,000, because if you gather more data, why to stop? But it's some international project that gather data from different ethnics. So you have your cohort, now it's time to do some association studies. So I already show you this abbreviation, and you will see it oftenly in some in genomics. It's called Genome-wide association studies. So Genome-wide means that you are looking to the whole genome, you are looking through the whole DNA of people. And association studies means that you try to find some association. Does that variant somehow associate with the phenotype or not? So you usually test single nucleotide polymorphism, and I will talk about it in a little bit more in a moment. It's some variable place. So maybe you know from some newspaper articles that 99% of our DNA is the same. So it's that 1% or less than 1% that we're looking into. You want also to look on the common variants where the minor allele frequency, and allele is a form of that variable, and we'll look into that in a moment, it's more than a 5%. Why so? It's just because to make it statistically significant. So in case you're interested in some other type of analysis and you're eager into, you can Google rare variant testing, but I will not talk about it today. And so, as I said, all of this is like a bunch of regression models. So GWAS in its essence is like linear or logistic regression based on what phenotype you are using. So if you are using binary phenotype, it's a logistic regression. If you're using continuous phenotype, it's a linear regression most of the time. The interesting part comes in a GWAS. It's not about the method itself, but how to make it scalable. So I was talking about UK Biobank where we have 500,000 samples. Now our company will be providing data for Our Future Health Foundation. It's also in UK, and they plan to have 5 million people. So it's not now a problem of bioinformatics, it's more a scalability problem. So you have 5 million samples. How will you do linear regression on that, on this like very dense data? Because you have 5 million samples, and each of the sample will have several million variables you are looking into. So this is the actual problem that we're having right now in a genomics. And in case you want to look into what are the different GWAS studies already, you can look into GWAS catalog. It's an open catalog for curated GWAS studies you can look into. So I was saying that we are looking into variable regions. So what actually variable regions are? So this is our DNA. We have to make a double helix because, you know, DNA has to be a double helix. So let's say that here is a single nucleotide polymorphism. So some place where some people has, let's say, letter A, and another people has letter G. So here you see that different letters in the same position are called alleles. And going back to this slide, having minor allele frequency, this is what it is. So if one of it is like less frequent, you want the less frequent to be at least 5% to make any conclusions for your study. So here is a schematic look how the GWAS look like. So for the binary phenotype. So you have normal samples, disease samples, or any samples with your phenotype, not especially disease. You take DNA, you make some analysis, and then you look at the frequency. And based on that frequency, you're saying, oh, that region is important or that region is not important. So this is the typical, if you want to dive into GWAS and you want to know like how the results look like, so this is a typical GWAS plot. It's called Manhattan because this looks like a skyscraper. Okay, so on the x-axis we have chromosome, and on the y-axis we have p-value. It's a negative logarithm of p-value. So you see that like those, and here is some threshold, significance threshold, and these are the regions that you would look into. Like these are significantly associated regions with the disease or with any phenotype. So in this case, it's a diabetes. So these are the two genes, one on a chromosome 7, another is on a chromosome 10, that are associated with that disease, meaning that people that are having specific alleles in that genes will more likely have or have like increased risk of diabetes of type 2. So we're in a GWAS article and we want to look like how the results look like. So we saw the plot, and then for this, like for some selected markers for selected genes, you will see this table. So here you see that there's that gene that I was showing you before. The two genes, they have some they are on a chromosome and the position. They also have a specific ID in a database. So here we see, and you sometimes can find some articles that this RSID is associated with that. So the RSID is coming from DB-SNP database, again, another curated database for variable regions. So these are SNPs. We know about their position, and here we see affect allele. So this is an allele that is somehow connected with the disease that we are studying. Nearest gene, because it might not be in the gene region. Maybe you know something about junk DNA, which is not junk anymore. So about 20% of our DNA is actually protein-coding genes, and the rest is not known. There are some like non-functional genes, or some like remaining part of virus of our ancestor had before. Like, it might be junk and might be not junk, because there are some regulating parts there. So that's why you have the nearest gene. You also have an allele frequency here. So see that this allele is very frequent. 90% of the people has it. And here we have actually our effect. Sometimes it's as an effect here in that publication, it's an odd ratio, meaning that if it's more than 1, it's likely that the person with that allele will have that trait. In this case, it's diabetes. And here is a p-value. So these are the results that we're having for the association studies. Now that we got it, we're moving to our next stage, and it's polygenic risk score. So what polygenic risk score is, it tries to estimate a person's risk for a particular disease based on what variants person has. And it, as I said, like, again, it's regression, it's a penalized regression, and the data you are starting with are the data that I show you here. So once you selected significant variants from here, significant SNPs, you are then using a polygenic risk score to actually go, like, analyze each sample and say what is the risk for that person. And again, in case you want to dive deep into that, you can look into polygenic risk score catalog, where there is a curated list of like thousands of different published polygenic risk scores, and also there is validation on a different samples. So I wanted to show you how the usual result from the polygenic risk score study would look like. You would have RSID that we know from the before. It's some ID of that variant. We have an effect allele, and then we have effect weight. So it's weight over the regression model, the usual regression model that you would use elsewhere. So now that we have our polygenic risk score, and we want to apply it, it's relative. So it's not like if you have polygenic risk score of 10, that means that you have disease. Or if you have on, I don't know, like -2, that means that you don't have to go to the doctors at all. It's all based on comparison with all of the other people in the population. So what you do is you calculate polygenic risk score on an appropriate population for the score distribution. Then you calculate for that individual, and you look where it sits in the distribution. And based on that, you can look into percentile and define risk for that particular person. So usually it would look like this. So most of the people have some average risk. And I really, in case you like polygenic risk score, I really encourage you to look into polygenicrisk-scores.org, where they have really very nice animation how they're explaining it. So most people will have average risk. There will be some lucky ones that have decreased risk, and some of the concern with increased risk. And so in case the person is in 95th percentile, we could say, well, maybe you need to go to the doctor more often, and then it depends on the collaboration with clinicians, how they defined like what that person should do, or that person is lucky, it's like in 5%. It's like, that's awesome. Maybe you should just follow general rules for prevention for that disease. And here I just show you a teaser for our example that I will show you in a moment. So this is actually what you do. You first select the phenotype, you make association study, you pull the significant parts of genomes, and then you make a model to figure out what is the risk for each of the sample to have that phenotype. So we'll now look into the code. So the code is already published on GitHub, and you can run it in case you have your DNA data. So let me ask you, how many of you sent your data to DNA company, whatever, for ancestry analysis and so on? Okay, so there are a few of those. So you can start right away. And for those who don't like, you can today make an application and start playing with it. So those who are in the back, could you see it, or should I increase the font? Okay. 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 So here is some description, what we will be using. So my husband, Philip, was so kind to provide his data for this demonstration. And we'll be using as an input his data, which are obviously not published, you should use your own. And we'll be using a polygenic score from the polygenic catalog. And the output will be the image that I showed you with a percentile for the disease. So we will be using the selected polygenic score. How did I select it? Well, it was a very hard decision, but I'll show you in a moment. So our data come from MyHeritage, and we need to tweak them a little bit. So I exported this data, and then I tweak it so they are in an appropriate format. Then I spent a lot of time trying to find some score that matches with the threshold of variance, because the data was for ancestry and it was not for medical use. So that's why you see that there is an error, and it's like 8%, 9, 37, and so on. So because it's just basic ancestry analysis, and these are all of the medical traits. But I was lucky to find some for diabetes, and that's why I selected this specific score. So I selected the specific score, and here is a score file. So as you saw it before, RSID, affect allele, and this affect weight. But because I said that it's relative, I need to find some data and actually to make the distribution on that data to figure out, like, is that score, like particular score for Philip, is it like high or is it low? So first I made prediction on his data, and then I went to 1,000 genomes, and I downloaded the data for the chromosome that I wanted. So polygenic rescore in this case was only in some chromosomes, so it's 6, 8, 9, 20, and so on. So I downloaded only these chromosomes from the FTP server, and and then what I did is I actually need to now compute polygenic risk score on this data that I downloaded from 1,000 genome. So here is like a lot of like mingling. So how do you, like, now I am extracting this RSID from the VCF files and its variant call format files. And you can read about it in the details in the notebook. I try to make it like as descriptive as I can. So I extract it, and then I'm merging all of this VCF file into one, and then I am moving it into the table. So after all of this like different mingling, I'm ended up with a table. Yes, with the two tables. One is a table with the actual genotypes. So here are identifier of these people, and another is, and it's part of the data format that is used in bioinformatics, is information about the variable regions. So it's chromosome and position number. And then it's really, as you see, it's just Pandas. So it's loading as a data frame, then counting affect alleles. based on what people, exact allele that sample has, and it's done here, and then plotting it. So then you just use histogram to plot all of the individual polygenic risk score that I counted here. So the result table that I will be plotting into will be the one record per each sample, and these are the variables or variants that are used in that polygenic risk score that I selected. So once I selected it, I can plot the value for the Philip, and thankfully, I see that Philip, you are only in 72nd percentile, so it's okay for you. You should relax. So yeah, hopefully you were inspired by this talk. I actually forgot that I have one thing then. So you might think that this is awesome, so what should you do next? So you should learn bioinformatics. Rosalind is a perfect way how to learn bioinformatics with Python. I really encourage you to look into that. Those of you who didn't raise your hand for the DNA data, you should go and sequence your DNA. And you should definitely visit my husband's talk about APIs, because family business is family business. So I definitely encourage you to visit Philip Sedlák talk at 3 p.m. today. And thank you for your attention.
Anastázie, thank you so much. It was a great presentation. I do have questions for you.
Okay.
From Vojta, perfect presentation on an interesting topic. How common is this? Is it used routinely for masses of patients, or is it rare or experimental approach?
Yeah, so I am not sure about Czech Republic, but I know that in UK, since it's really like going into the sequencing mode, there is several startups that are having it as a bonus for employees. So like you have this program, for example, for heart diseases, because the heart diseases are one of the major cause of premature death, so like for people like in young age. So you could have this prevention. So I think that in Czech Republic it's just starting, but in UK and US, I think that there are like already companies that are doing it and are profiting.
Perfect, thank you. Another question from Matej. I am afraid of sending my DNA to MyHeritage. Is there any other way? How can I obtain my DNA data?
Yeah, sure. You can send it to China, for example. So, because most of the time, U.S. Companies are also sending it to sequencing. So, what you can do in case you are afraid of doing that, you can collaborate with some DNA sequencing laboratories here in Czech Republic. So, instead of having it like a study for you, you can just have a routinely sequence it as a normal lab that doesn't have sequencing machine would do that. So instead of like contacting Ancestry, like some startup, you can contact the DNA sequencing facility. However, your data still be there, or you could buy Oxford Nanopore, which is a sequenator that you can plug into USB, but you need to make a lab around it, or you can borrow some labs. So you can make creative with that. But if you don't want, if you want to generate data yourself, definitely check Oxford Nanopore.
Well, if you're going to plan to do a lab, share it on Discord so maybe people can join you.
Yeah.
Okay. And the last question here, it's more futuristic. Hello, Anastázie. Is there anything you are looking forward to that you think future can bring in this field?
So what I'm looking into, definitely not like, as people are saying, like select whether your child will be blue eye or like, I don't know, like genius and so on. What I'm looking more is to actually pharmacogenomics, which is not so common in Czech Republic, definitely. It's only used in for children who need to receive treatment. So they're analyzing their mitochondrial DNA and they understand whether they are fast metabolizers, that means that they need to have bigger dosage of the treatment, or there are low metabolizers, that means that they need to lower dosage. And this is the thing, and I have some cases in my family where this could ended up in person decided not to pursue treatment because it was just toxic, because that person is a low, slow metabolizer. So I'm looking forward on routinely making pharmacogenomics into our general practitioner labs or offices.
That's awesome. I'm looking forward to that too. In the meantime, one more question. Adela is asking, is it possible to predict a probability of higher disease risk in a child from the parents' data?
Yes, definitely. This is what medical genetics is doing by taking parents' data and combine. But it will be there should be another probability. So it will be a probability of disease multiplied by probability that that specific genotype will be. So it will be not so certain, but it definitely is a possibility to do that.
Okay. Thank you, everybody, for your questions. Thank you for your answers and your talk.
Thank you.